Hello, my name is Audrey Lapendry. I am a third-year PhD student in bioinformatics at the ENS de Lyon, in the laboratoire de biologie et modélisation de la cellule. With my colleagues, we are working on the impact of compositional biases on the 3D organization of genomes. We work on a project called Compositional Biases Promoting Self-Assembly, establish a link between the genome and the cell-spatial self-organization. To start our project, we use two types of data. The first one, IC data, to define a spatial cluster of genes, and the second one, GPSeq data, to determine the radial position of a genomic region. With this, we show that the spatial clusters are composed of AT-rich genes at the nucleus periphery and GC-rich genes at the nucleus center. Then we obtain the affiliation of genomic regions through compartments thanks to the spin datasets. We demonstrate that the genes of the lamina are rich in AT, while those of the speckle are rich in GC. So our model is that colocalized genes in the nuclear space share the same compositional biases. Then, thanks to list of activated genes by transcription factors, we show that some transcription factors activate AT-rich genes at the nucleus periphery, while others activate GC-rich genes at the center. So our model is that colocalized genes are regulated by the same transcription factors. Next, since colocalized genes have the same compositional biases, we then test and then demonstrate that they produce pre-mRNAs and mRNAs that also have the same compositional biases. Then we wonder if those RNAs are regulated by RBPs such as splicing factors, in the same way that we demonstrate that genes are regulated by transcription factors. Thanks to the analysis of RNA-seq data, it also supports that compositionally biased RNAs are processed by the same splicing factors. Next, our data suggests that compositionally biased RNAs produce compositionally biased proteins that share the same physical-chemical properties. Finally, we reveal that colocalized genes produce proteins that share the same cellular space. In conclusion, our model suggests that there is a link between the compositional biases of genes, RNAs, proteins, and thus this impacts their different physical-chemical properties and their localizations. So the localizations of genes and proteins depend on their compositional biases. A consequence of this is that there will be an impact of the cellular spatial organization on the spatial organization of the genomes. So thank you. I am available to answer your question or give you more details.